Thank you very much. So we will be giving a bit of feedback on some of the achievements that we have made as comp in the last year, in 2022. We have only a very short time, I understand, 7 minutes, so I will be flying over my part. I know sometimes it will not make sense if you miss the regulatory science background, but I guess afterwards questions can be asked if it's necessary, and then the second part will be done by my good friend and colleague Elizabeth here. So we'll start. I think the first thing that we want to highlight is something near and dear to my heart, to my own heart, namely ophthalmology and very specifically inherited retinal dystrophies. So last year, after about 5-6 years of being in the woods, the comp has finally come down with a new ontology for IRDs. So how that we will define conditions within IRDs for orphan designation moving forward. So very quickly, what is the problem with IRDs? So if you don't know, or if you know, of course, IRDs, they are what essentially a spectrum disease. So it is a huge group of genetic etiologies that have a very varied expression on a phenotypical level. And these expressions can be, I mean, even within the same gene, if you're in a family, children with the same gene defect can have very different expressions depending on which specific locus is affected, which modifier genes are involved. And now, a big issue with this is that a lot of what we still see today in science literature is that we are still talking about these diseases in a what I call the classic classification, so in a phenotypically driven classification. So we talk about retinitis pigmentosa, Leber's congenital amaurosis, etc., etc. And these were all names given at a time when people didn't understand the underlying genetic etiology. Now, as you can see on this, next slide. Sorry, I should say next slide. Next slide. Apologies, I'm not controlling myself. So as you can see here, this is the bubble which I keep bringing around for 5 years now, which kind of shows you all these little black things. I know it's not very legible at this size, but all these black things in the bubbles are genes. It's very outdated, by the way, because we have now around 345 genes known that cause IRDs. And then the actual soap bubbles or the colored bubbles are these classical named diseases, congenital static night blindness, LCA, Ahlstrom syndrome, etc., etc. And you see how much that these overlap and where the same gene can give rise to very different classical diseases. Next slide, please. Okay, so this actually brought issues on a regulatory level for us. And here it becomes a bit difficult, but there is a bit of a divide between what we at COMP give as an orphan indication or an orphan condition and what a therapeutic indication is, which is what is given by the CHMP at the time of marketing authorization. And to put it very simply, a therapeutic indication can never fall outside of an orphan indication. So an orphan indication should always be as broad or broader than a therapeutic indication by law. Now, in 2018, a product came to the market, Luxturna, and here we had a bit of a problem. And you can go to the next slide, please. No, sorry, it's another thing. But the problem here was that Luxturna, when they came to the COMP, they received orphan designations for the treatment of retinitis pigmentosa and LCA. But at the time when they passed by CAT and CHMP, they were actually told, because Luxturna is a viral vector that contains a functional copy of the RP65 gene. They were told by CAT and CHMP, no, your indication should actually be treatment of IRDs caused by mutations in the RP65 mutation, which is scientifically the most correct, because that's what they're doing. Now, the problem there is that when it comes to IRDs, our knowledge keeps growing and growing every day. And we now, for example, know that one of the diseases caused by RP65 mutations is also retinitis pigmentosa with choroidal involvement. Now, despite that it is called retinitis pigmentosa, you can make a very strong argument that it is actually a chorioretinal dystrophy, which is very distinct from typical retinitis pigmentosa. And now the problem, of course, is since they had orphan designations for retinitis pigmentosa and Leber's congenital amaurosis, this new disease that they can treat with their product does no longer fall within the orphan indication. And technically that would mean that they lose the orphan indication. Now, in this particular case, it was covered with a mantle of love. And we said, okay, there's retinitis pigmentosa in the name, it's fine, it's covered. But it was kind of an indication that in the future, this will happen more and more, especially because genetic treatments are becoming the de facto norm for these IRDs. So we knew we had to do something because our classic phenotypically driven ontology was no longer tenable in the face of scientific reality. And so we started working on this. And in the beginning, we kind of went all ways. I mean, it was very bad. We tried all kinds of things. We gave designations that were not very continuous, that were bad. So in 2021, I actually thought we should really start working on coming to an ontology that is usable and that is fairly robust moving forward. So I was lucky to acquire a book at that time where the authors used a model that I thought would be quite useful to use for us as our orphan designations also. So in 2021, I presented a proposal to COMP. They were all very enthusiastic about this. We also went to the COMP CAT meeting. When we had a meeting together, we also came to CAT, and everybody at CAT was also very happy with that. Now, the problem with that I had is that I think a ID that does not get pushback is not a good idea, because it means that either people do not understand the ID or people are not interested. And I don't think the people are not interested, so I think it was mostly that we lack a lot of ophthalmological expertise in the committees, or at least the committees where I went to explain my my model. So we then at COMP decided, okay, after I pushed for it, that we will form a working group for this, and we started in early 22, where we start to do refinement of my proposal, and very quickly we start making holes in the model that I proposed, and we very quickly also saw that we were not going to be able to do this by ourselves. So we start preparing and then effectuating an expert meeting in June 2022, which involved both clinical experts in Europe and patient experts on IRDs to see where we should go with this model and what would be a good thing for us to move forward. That went actually very well, a lot better than I'd hoped actually. We had a lot of good discussion also afterwards, still with the experts and the patients. And so we had our final proposal end of last year of the new model that we will be using. And this was now accepted by COMP in January 2023 officially, and it has now also been published on the EMA website. So next slide, please. And I apologize for the very small letters here because this is a screenshot from the proposal. But what will happen now is that IRDs that now come to COMP, instead of going with, treatment of retinitis pigmentosa, there will now be 3 possibilities. If you come with a gene therapy that's very specific, you will just get inherited retinal dystrophies caused by dysfunction of your target gene. The other thing that you have to keep in mind, there will also still be products that are very broad, that do not target just a specific gene. Now for them, you see in this table 1, we now have a new model that we will use for giving the designations, which is based on my initial proposal, but you don't see much of my initial proposal in there anymore. Unfortunately, it's been very much changed. So, and basically it will boil down to, are you a syndromic IRD? So are you associated with a syndrome like Joubert syndrome or Ehlstrom syndrome? Or are you a sort of standalone IRD that is not associated with a syndrome? And then in there, we will look, are you cone-driven, which means are the cones first affected? Are you rod-driven? Are the rods first affected? And then macular dystrophy, which is a bit of a catch-all term for everything that doesn't fit in the other 2 subcategories. We have the choroidal dystrophy, so the dystrophy that involve the capillaries behind your retina. And then last but not least, we also have the vitreoretinopathies, which are those that involve your vitreous body of the eye. So this is a model for every treatment that could treat more than one of these IRDs or more that goes over genes. And then last but not least, there will still be a few diseases that do not fit in either of these. And for these, we will still foresee the possibility to have a very specific designation. And I think, for example, X-linked retinoschisis is one of those. And now you may say, oh, this looks all very logical, but I can assure you it took a lot of time and effort to come to this, to come to a model that we feel that will also hopefully stand up to the test of time, because this is a field that is in constant motion. And now that we have kind of a template on how we did this, this will now also be used by COMP in the future moving forward for a lot of other conditions where we know that they are actually genetic spectrum diseases rather than specific conditions. And we will use this now also to move forward to change a lot of ontologies in the future, I think. So thank you. And then my colleague will take over, sorry, for the second part.
Right, it's very short. But before I give you a list of the products, orphan medicine products that are authorized last year, next slide, please. Just a quick overview how orphan designation is decided on by the COMP. Well, first, the COMP decided very early in development, drug development, might even be based on non-clinical data or very preliminary clinical data, and then they have to decide on the condition. It might be a challenge on itself, because it's before the drug development continues in the marketing authorization for the final therapeutic indication as established by the CHMP. So in the first instance, whether it's rare, and also in the initial orphan designation, the COMP look whether there's a potential for significant benefit for what's already available for the treatment of that disease. And why is that so done early? That's because there are incentives for orphan designated products. That there is no fee for academia or small and medium enterprises, or low fee for the big companies, for protocol assistance, and the fees for the if they come through all this process for drug development, if they are successful, for marketing authorization applications, because there's a subsidiary from the European Community. Then, of course, marketing authorization is decided by the CHMP. They decide on the benefit risk and the final therapeutic indication for the drug, mostly on its own, not necessarily compared to other treatments. But for the orphan designations, that might be years in between in this process. We have to check again, does it still fulfill the criteria of significant benefit? And because there might have been changes in treatment paradigm in all those years that drugs in the development, other treatments may have been approved, or other methods. Next slide, please. So last year there were 24 products, orphan medicine products authorized by the CHMP, and also got the final orphan designation, the maintenance of that, by the COMP. 24 of 89, so it's quite a lot, and I think it's about 25 to 30%, and it's still increasing interest. So that's a good thing for the rare disease community. And this is a very broad therapeutic range, might be from rare cancers to hereditary disorders, and I think Mencia discussed upstasa, but there are also for the epidermis bullosa blistering disease, and I think the patient input was very important here at the CHMP. Next slide. I can discuss them all in this short time frame. Sorry, I would love to do that, but there's no time for that. But it might be, maybe you're not aware of, it can also be the situation that on itself, the benefit risk is positive for a product, but it's not significant better than anything else that's authorized in meantime. And that happened to 6 products last year. And so they are authorized. You can use it, and they are effective, and they outweigh the safety is acceptable, at least. But it mostly happens in the hemato-oncology field, because we got so many products, so it's difficult to establish whether they're really superior to the other ones. So and that happened to 3 products. Diffuse large B-cell lymphoma, we got, I think, about 6 or 8 products in the last year. So it's more and more becoming more challenging to see whether it's really, really better. And other products, the enzyme replacement therapy in Pompe disease. We have Myozyme, and an attempt was made by a difference in the utilization of this product. to make it less vulnerable for drug antibody formation, and to be more effective in the target, which is the muscle strength. But they failed to demonstrate so. It's very difficult. It's a rare disease and a very heterogeneous disease, as most of these orphan diseases are. And in the end, the studies, it could not be decided whether it was really superior. And even there was a discussion whether this could be considered as new chemical substances. So these are marketed, but are at least marketing authorization, but not anymore as an orphan product. So they don't have the extensive protection of 10 years, but the regular protection as for any other product approved by the CGP. Thank you.